So hello guys, today we are conducting an interview with Dr. Katherine Frels. And first of all, doctor, it's a pleasure to have you here in our event. And I'd like to take you some questions so we can interact a little bit. So what motivates you to pursue a scientific career, particularly in the field of small grain breeding? And how did you train your this in your during your graduation and post-graduation?
Thank you. It's a real pleasure to be in Brazil for the first time and to be joining you at this event. I've enjoyed it very much so far. And so I have always been interested in science, and I liked plant science more than other sciences. And I was lucky enough that when I was in high school, that I was able to take a biotechnology class. And that's where I learned that all of those corn and soybean fields that surrounded my home. I'm from Iowa, the land of all of the seed companies, right? Pioneer Hybrid and Monsanto, all of them have large locations in Iowa. But even though I had heard of these companies, I didn't know what they did. And it was that biotechnology class that made me realize that someone actually had to go develop all of these different corn and soybean varieties that I could see. And so that was really fascinating to me. And I visited Iowa State University, learned about plant breeding, and realized that is the career. So I did my undergraduate degree at Iowa State. I did a PhD in plant breeding and genetics at the University of Nebraska. And that was in small grains breeding and genetics. I went then on to the University of Minnesota and worked in alternative crops development. And so my career track has always been focused on plant breeding, but I've always made sure to try to work with different species, different people to gain more experience. So I went from corn and soybeans in Iowa to small grains in Nebraska and then to alternative crops in Minnesota. And that gave me a very wide range of experiences and helped me get where I am today.
Oh, really nice. And what do you think is the main challenges you face when you are applying transcriptomes in this small grain breeding? Like when you consider the complexity of these crops and mainly the interaction of genotypes and the environment.
That is a huge problem with dealing with a polyploid species, especially one with a very large genome. It is more expensive for us to get coverage on any marker platform, whether we're talking about a genome-wide sequencing effort or transcriptomics. If we want to have enough coverage in our data sets, we have to do fewer samples than maybe a diploid crop or a crop with a much smaller genome. So fewer samples, which means maybe fewer environments, or we pay a lot more. So that can be a significant challenge when we're talking about the sequencing depth for any type of data platform. So I think that is a limitation for us, especially if we're trying to study how a trait reacts in different environments. But at least for me as a plant breeder, I get to partner with a lot of plant biologists, geneticists who do more of that research. So transcriptomics is a little more up the basic research upstream from me. I'm very downstream in applied plant breeding. So most of my work with transcriptomics is going to come through a partnership with someone else.
Oh, really nice. And talking more about that partnership, like in your research you use to study gene expression, what are the most significant insights you have gained regarding key agronomic traits, such as disease resistance and grain quality, things like that?
So that, again, is a little bit of a challenging question for me because I think the insights that we get through transcriptomics are more about how things are changing within the plant. And as an applied plant breeder, I am most concerned with the end product, right? So that final grain yield, because that's what my stakeholders care about. My stakeholders are the farmers, they are the millers and the bakers that are going to purchase wheat from those farmers or from the co-ops that buy from the farmers. And so it can be very challenging when we're thinking about doing some of the discovery that is associated with transcriptomics to see how it's actually going to apply into my plant breeding program, because I am mostly worried about, is that phenotype there or not necessarily how it comes into being. So I think transcriptomics is good for discovery, but it takes a lot of validation and understanding how we can potentially transform this from looking at the plant response into a marker that I can use very quickly in my breeding program and apply it to selection.
Oh, really nice. And I realized that you are a very really attached person to quantitative genetics.
Yes.
So I'm like this as well, and I think that for the main purpose of the breeding, like yield and things like that, we can never abandon the
Absolutely.
quantitative genetics. But looking for the other sides, like our event talking about omics, how you think like omics, like such phenotyping, transcriptomic, and other omics can be put in this kind of pipeline of a breeding program to enhancing gains even more?
That's a great question. I think all of the omics are most powerful when we combine them. So having your transcriptomic results without having paired phenomic results, it can be very challenging to understand, okay, we have got this biological process going on here, but what does it actually mean in the field? And then, layering genomics on that, which is what is easiest for a plant breeder to do, especially a plant breeder like me. I work in winter wheat. So we harvest in July, and then we have about 6 weeks to turn around, make all of our selections before we have to plant again. So it's a very short selection time. And that's what I mean by saying I need to go from those transcriptomic results into something that I can use very quickly. And so when we pair it all together, I think we end up with a better understanding of what the reality is. The other thing is a lot of our transcriptomic studies often are started in maybe a single location or even a greenhouse, because it's so much easier to sample, right? And capture those plants at specific growth stages or capture a very specific tissue. But we have to remember that when our final varieties are grown, they're being grown by farmers in lots of different environments. So any way we can extend that understanding to those wide range of environments and apply our quantitative understanding of traits like yield, I think that's going to give us the most powerful results.
Oh, really nice. And what do you see about the future perspectives of transcriptomes, and particularly in your area, that is small grain breeding, and in this context of climate change? Because as we were talking about, like you do the transcriptomes, but in different environments it can have some issues. And how do you see the climate change is affecting that studies?
So that is something that I think we need to take better care about. It is a plant breeder's job to predict the future. So the varieties that I am developing right now in my breeding program aren't going to be out on a farmer's field for somewhere between 5 to 10 years, depending on what stage of the program we're talking about. So I'm actually breeding for a future environment. So I think that is somewhere that we can really apply or try to apply transcriptomic studies that are conducted in a greenhouse or under special circumstances to try and understand how that plant is responding. So for example, one of the major challenges we have in wheat, it is a C3 crop. It likes cool temperatures, especially cool night temperatures. Well, one of the things that we are seeing is that night temperatures are warming. And so we don't get as many of those cool nights where the plant gets to rest and recover. And those C3 species just don't deal with that stress as well as a C4 species. So understanding what is happening in the plant under those warm night scenarios is something that I think we can definitely utilize transcriptomics to learn from. And again, we have to use those transcriptomics results to see, are there things that we can actually select on? It is very challenging for us as a breeder to select on a whole genetic pathway. Let's say we just need to upregulate the entire thing. That's very difficult for us to do because there's so many pieces in that pathway. So trying to identify those parts of the pathway that can have the largest effect with, say, for example, the fewest number of genes that we actually need to change. That's going to give us the best results in the shortest amount of time, at least in my opinion.
Oh, really nice. And a last question that is really interesting, changing a little bit of subject. What advice you give for students and young researchers interested in pursuing a career in small grain breeding?
Well, I'm very biased because I think small grains breeding is one of the best places to be at, especially if you are interested in academia. There are great private industry small grains programs, but currently small grains breeding is one of the few crops that are large enough to have a real societal impact, but still have a very significant amount of public breeding programs. At least in the US, we are probably one of the few large hands-on active applied plant breeding programs left in crops. There's a few for soybeans around, almost no for corn, and then we have lots of other smaller crops. But if you want to see a large crop and see a large commodity breeding program, I think small grains are a great place to be. So that's my pitch for small grains. But I think, for students to be successful in plant breeding, small grains or any crop really, I think it's really important to remember that if you want to go run a breeding program anywhere in any crop, it's not just that you need to learn the science and you need to learn the quantitative genetics theory, the Mendelian genetics theory, statistical genetics, all of that. You also have to learn the logistics of a breeding program. And that is one of the hardest things to learn if you don't have access to a breeding program. So I encourage students, you want to see as many different breeding programs as you can, even if it's just an internship, because every breeder does things differently and they run their logistics a little bit differently. And so then when you go take your position, you've seen all of these different breeding programs and you can turn your breeding program into something that works for you, taking pieces from each of those programs that you've seen. But the most important thing for an applied plant breeder is to get the right seed in the right packet, in the right plot, with the right genotypic information, and to do that very quickly and efficiently and to make your selections. And that logistics part is one of the things that I see students who who are coming out of a more basic science program, if they take a breeding position, that is the hardest thing for them to learn. So if you're in one of those basic science labs, I highly encourage students, if you're interested in working in a breeding program, make sure you have a committee member with a breeding program, or you have a co-advisor, or you do an internship. You want to see what it takes to run the logistics of that program. You can apply your science anyway, anyhow, anytime, but learning that logistics, if you learn that ahead of time for your position, you're going to have a huge step up.
Thank you so much, Doctor. Your presence here is very important for us, and I hope you are enjoying your time here in Piracicaba.
Thank you very much for having me.